Hi, my name is Sabrina Templo. Today is June 11th, 2020. This video is for Bio 225, Week 5, Lab Results and Reflection for Carbohydrate Fermentation Testing. It has now been a little over 12 hours of incubation, so carbohydrate fermentation testing is another form of biochemical testing. Biochemical testing is actually another type of test that scientists can use to help determine what microbe or bacteria they are working with, and it is actually more cost-friendly and I believe is more efficient than DNA sequencing, which can be challenging. The biochemical tests can allow us to manipulate different bacteria depending on which test we use, and it can help us determine what we're looking at and help us with further testing. So the carbohydrate fermentation is another variation of chemical testing. The fermentation is what actually happens to carbohydrates when they no longer have oxygen. So this is something that is anaerobic. The carbohydrate testing, we use 3 different powders, which is glucose, fructose, and mannitol. Glucose is a carbohydrate that is produced from plants and animals through photosynthesis, as well as humans and their metabolism and glycogen. And then the next one we used was fructose. It's a carbohydrate that is found in fruits and vegetables. And then finally we use mannitol. Mannitol is more of a crystallized alcohol and is produced by plants. Here I have my 6 tubes. I did my experiment and I used my S. epidermidis with glucose, fructose, and mannitol, as well as the S. cerevisiae of glucose, fructose, and mannitol. So basically, some of the final result products of mannitol is lactic acid, and an end product of fructose would be acetic acid or carbon dioxide. And glucose would have hydrogen gas, lactic acid, and ethanol. So those are just a few examples of some of the end products of fermentation with those substances. So basically, what we're looking for is during this experiment was to determine what fermented in those microbes that we have. And if the broth stayed red, it would indicate that no fermentation had occurred. If it turns yellow, it means that fermentation did occur with gas. And if it turns orange, it means that fermentation did occur, but there would be no gas. So here we can start with my yellow tubes. So I have my S. cerevisiae tubes. Here is my glucose, my fructose, and my mannitol. All 3 of these tubes came back with results for testing as fermenters with gas production. What happened with these is that they fermented, and obviously we can see that they did because they are clearly yellow in color. And I found it interesting that I noticed that after I checked it after 12 hours, these were already very yellow in color. So next we have my S. epidermidis, and I found it interesting that the phenol red broth is not the same as it originally was prior to this experiment. It's so much lighter in color. I'm not sure if you could tell from my camera and lighting. And I do understand that the oxidation of sugars occur, but I feel like these are more orange than red. So again, here's my glucose. Oh, sorry. This is my glucose, my fructose, and my mannitol, which is more orange in color than red. Like I said, I checked this over a little over 12 hours and it looked like the color had changed. And after a couple more hours, it seems like they lightened in color even more. I will say that I had quite a bit of difficulty with transferring the powders into the vials. So there may be a bit of error on my end, but I'm gonna go according to my results. And according to my results, that I'll say that my glucose, fructose, and my mannitol on my S cerevisiae are yellow, so they're fermenters with gas production. And my glucose, fructose, and mannitol on my S. epidermidis have somewhat of an orange color. It's certainly not a perfect orange, but certainly not a perfect red. But I'm going to say that they are also fermenters without gas. And I also had quite a bit of anxiety doing this experiment, making sure I transferred half of the powders properly, making sure that I was doing my aseptic technique, making sure that there was no bubbles in the Durham tubes, as well as having the Durham tubes dry, because it did take quite a while to it to dry from being inside the alcohol. And also I did find it interesting that everything changed in just a span of hours, and it was very quick. So that concludes my video for carbohydrate fermentation. Thank you.